AP 120, Chapter 4, the topic's DNA replication and transcription. All right, you should know what this picture is. This is DNA, our deoxyribonucleic acid. And as we know, it is a double-stranded helix. DNA is where we store all of our genetic information. The information is in little packets or chunks of DNA referred to as genes. And each gene will lead to the production of a single protein. So we have lots and lots of genes on a chromosome. Each gene is just a piece of DNA, and those genes can lead to the formation of a protein. A genome, that term means all of the instructions, all of the DNA in an organism. All right, how many chromosomes does the average human have? It's 46. 46 chromosomes coming in 23 pairs. 23 pairs of chromosomes, 46. Here is a karyotype. This is where they take a dividing cell and they are able to actually squash it and examine the chromosomes, pair them up to see if the person has a normal or abnormal karyotype. So this image, is this person male or female? They are male. We know this because the sex chromosomes, which is the last pair, is XY. If it was female, it would be XX. So is there anything else unusual about this karyotype? That's right. There are 3 chromosome 21s, trisomy 21. So this individual is a male who has Down syndrome. All right, the nucleotide is the monomer for nucleic acids. And nucleotide has a phosphate group, a 5-carbon sugar, and a base. So remember that. 3 components: phosphate group, 5-carbon sugar, either deoxyribose or ribose, and nitrogenous base. You form long chains of these to get DNA or RNA. So when we look at DNA, we see that it is a long chain, and the chains are formed via phosphate and sugar covalently bonding to each other. So go sugar phosphate, covalent bond, sugar phosphate, covalent bond, and so on. So that's the sugar phosphate backbone, represented by this strip in the cartoon model. And then the bases, the nitrogen bases, point out, or in this case, inside the double helix. So you have bases pointing in, attached to each other via hydrogen bonding. That's how we get the double helix, the 2 strands connected together. DNA is 4 nucleotides that it uses: ACGT, adenine, cytosine, guanine, thymine, and they complementary base pair in a very specific manner. Adenine will only bind to blank, and cytosine will only bind to blanks. We look over here, cytosine only binds to G, guanine. Adenine only binds to T, thymine. And of course the reverse, T binds to A binds to T, C to G, G to C. And this is how complementary base pairing works in DNA. So here is a lovely strand of DNA. A will bind to, C will bind to, anyway, A will bind to, G will bind to, T will bind to, A will bind to, C will bind to, G will bind to. Hopefully you got the answers because I told you how to do it. All right. The other thing worth noting in the double helix is the 2 strands of DNA are oriented or pointing in opposite directions. All right, DNA replication, the production of new DNA. As we know, this occurs during the S phase of interphase. And we start with the original DNA. The original DNA is then unwound, so the double helix is split, forming 2 single strands of DNA. And then the new strand of DNA is formed on the old strand via complementary base pairing. So if there's a T in the old strand, you get an A in the new strand, C in the old strand, G is added in the new strand. And this is done twice for each of the single-stranded strands of DNA formed from the original DNA molecule. So each old strand acts as a template for the new strand. There are some key proteins to this process. There is helicase. Helicase protein unwinds the double-stranded DNA. And then we also have DNA polymerase. This enzyme complex, DNA polymerase, produces the new DNA. It reads the sequence and brings in the complementary DNA nucleotides. Two sets of enzymes that are very important. So we need, to do DNA replication, we need the original DNA. We need DNA nucleotides, we need helicase, and we need the DNA polymerase. Now, DNA replication is not a perfect process. Mistakes happen. Sometimes there are mutations. The wrong nucleotide gets brought in and added to the growing chain. Overall, though, it's extremely effective, but again, sometimes mistakes happen. All right, DNA, as we know, stays in the nucleus because that is the safest place for the DNA. We don't want to risk it being damaged or lost in any way because it's critical information stored there. So that information for a single gene needs to leave the nucleus to get into the cytoplasm where ribosomes can be used to make proteins. So what carries that information for a single gene out of the nucleus? Options: mRNA, rRNA, tRNA, or DNA. It is mRNA, messenger RNA. It's carrying the message. It's copying one gene and carrying that information out to the cytoplasm. The process of making the messenger RNA is called transcription. Transcription. And this occurs in the nucleus. Basically, an RNA polymerase enzyme comes in, it opens up the double strand, and it begins making a copy of that gene. So it makes a short single-stranded RNA using the DNA as a template. And in this process, it is, of course, complementary base pairing to the single-stranded DNA. And it's also worth noting that in RNA, you have uracil instead of thymine. So anywhere in the DNA that you have an A, the complementary base pair in the RNA would be U, uracil. So here is a DNA sequence. What would be the messenger RNA sequence? A would give us, G would give us, C would give us, G would give us, T would give us, A would give us, C would give us. Hopefully you got it correct. A's in the DNA leads to complementary base pairing and U's in the RNA, G's to C's, and so on. So complementary base pairing similar, except we have U's instead of T's. All right, so we have that messenger RNA copy of a single gene produced. What did genes code for? Hope you said proteins. Messenger RNA made in the nucleus has to be in the nucleus because it needs the DNA as a template, and then the messenger RNA leaves the nucleus and goes into the cytoplasm where the ribosomes are. And that is it for this part of the chapter.